In order to make the proteins accessible to antibody detection, it must be transferred onto a membrane from the gel. Cut for polyphenolidene fluoride membrane, or PVDF, to fit the measurements of the gel. Every interaction with the membrane should be handled carefully with a rounded tweezer to avoid puncturing the surface, and always hold the edge of the membrane. Soak membrane in methanol for 30 seconds. Wash in water for 1 minute. Finally, place in transfer buffer until ready to use. Separate glass plates using a plate separator.
Okay, so you can see the gel.
Remove and discard the stacking gel using the plate separator.
Off the top.
Cut the corner of the gel in the latter column and soak in transfer buffer.
There.
Now create transfer sandwich.
Next time.
During this process, everything should be wet in the transfer buffer.
Go like 1 or 2 steps of the blotting. So this stuff that I'm moving over is important. But gel, okay. Like have the gel out and then have it roll. That's like 1 step, right? So show the important steps. Have the gel out.
Roll out the gel gently to remove any air bubbles, as they will affect the protein transfer.
And then step 3 is like get your membrane and lay it on top.
To ensure the proteins transfer properly, align the cut corner of the gel to the cut corner of the membrane. Lay the PVDF membrane on top of the gel and roll out any air bubbles.
So you show like after you put the membrane on top, roll it out.
After removing air bubbles, lay the filter pad on top of the membrane and finish the construction of the sandwich.
So roll it out one last time before taking it out, and fiber pad. Roll it out one last time.
Then, clamp the cassette together and slide the top hinge to close.
Okay, good. Transfer buffer. Show.
Fit the cassette inside the transblot central core, always match black to black, and place it in the electrophoresis chamber with transfer buffer and an ice pack before securing the lid.
So here I'll turn it over. It's like the ice pack's there to keep it cool. Yada yada yada. Put this on. So
Connect to power supply and run for 1 hour at 100 volts.
that should stop. And then on. You should be able to see the bubbles and just say that's how you know it's really working. Good.